Good day, everyone, and welcome. Thank you for joining us today for a webinar on a deep dive FDA draft guidance on statistical approaches to establishing bioequivalence. My name is Ray Ford, and I'm with the Small Business and Industry Assistance Program, which operates within the Division of Drug Information, which in turn resides in FDA's Center for Drug Evaluation and Research, Office of Communications. I'll serve as your host and moderator for today's webinar. For your convenience, this slide provides QR codes for our social media avenues. You can scan them now with your phones and be taken to the appropriate platform. Before we begin, I'd like to cover some details. Slides for today's presentation are available from the right-hand side of your screen, where the orange button says Download the Presentation. This activity is eligible for an attendance certificate, which supports continuing education by SOCRA, RAPS, SQA, and ACRP. Please refer to our website at fda.gov/CEDAR-SBIA for more details. You'll be able to obtain the attendance certificate only upon completion of the survey, which will remain open for 2 weeks. Please remember to download the certificate immediately upon completing the survey. It cannot be retrieved after the fact. This also means the survey will close on March the 28th. Even if you don't need the certificate, we would appreciate it if you let us know your comments and feedback via the survey. The webinar will be posted in its entirety on our website within 5 to 7 business days. And that website is fda.gov/CEDAR-SBIA Learn. Additional tutorials are available through fda.gov/CEDAR Learn. Please join us April 12th and 13th for the FDA Generic Drug Forum 2023, celebrating 10 years of the Generic Drug Forum. You'll notice that the title is hyperlinked to the registration page for your convenience, so please register for GDF 2023 right after this webinar. To outline today's event, the webinar in its entirety will run approximately 2 hours, which includes a Q&A session. And if you have questions now or during the webinar, please enter those questions into the Q&A chat pod. Please remember also to indicate at the beginning of each question the name of the speaker to whom the question is being addressed. We'll answer as many questions as time allows. This slide provides a snapshot of the entire lineup of great speakers that we have scheduled for you to hear from. Please click the Download Today's Agenda title at the top of this slide to download a detailed copy of the agenda as a handy reference to follow along with each presentation. To get us started, it's our pleasure to introduce Dr. Liang Zhao, who's been serving as the Director of the Division of Quantitative Methods and Modeling, Office of Research and Standards in the Office of Generic Drugs since 2015. Dr. Zhao has a broad spectrum of scientific and managerial experience in industry and regulatory agencies. Through his 16-year professional career, he's established his leadership in industrial R&D, quantitative methods and modeling, and model-based strategic decision-making in regulatory and industrial settings for generic and new drugs. He initially joined the FDA as a clinical pharmacology reviewer in the Office of Clinical Pharmacology in 2009 and worked as a team leader in the Division of Pharmacometrics from 2013 to 2015. Please join me to welcome Dr. Zhao to kick off today's presentation. Welcome, Dr. Zhao.
Hello, everyone. My name is Liang Zhao, Director for the Division of Quantitative Methods and Modeling. It is my great pleasure to kick off the deep dive to the FDA draft guidance on statistical approaches to establishing bioequivalence. I want to start from a famous quote from Isaac Newton. Newton was explaining that he built on the work of those who came before him. The draft guidance we present is the result of years of experiences in practice and knowledge accumulation from many scientists. It reflects the current status quo of understanding of using statistical approaches to establish BE. The FDA has been developing and updating its guidance on statistical approaches to establishing BE since 1992. The initial guidance focused on a standardized approach to BE studies using a standard two-treatment crossover design. In 1997, the FDA proposed using population and individual BE approaches to assess bioequivalence in a preliminary draft guidance. 2 years later, the FDA expanded on its guidance and proposed 3 different approaches for establishing BE. In 2001, the FDA released a more specific guidance with the same name as we are using today on how to conduct BE studies. In 2022, the FDA released the latest draft guidance incorporating new statistical methods and advances in technology. These guidelines ensure that BE studies are conducted consistently and reliably to ensure the safety and efficacy of generic drug products. As we know, the FDA has been developing a set of core guidances to provide recommendations on how to meet the provisions of Part 320 for all drug products. This guidance specifically focuses on the requirement of a BE and the appropriate in vitro and in vivo studies to measure BE and establish BE. Once finalized, the guidance will represent the FDA's current thinking on the statistical approaches used in BE studies. It is important to note that this guidance does not establish any rights for or on any person, and is not binding on the FDA or the public. However, alternative approaches may be used if they satisfy the requirements of the applicable statutes and regulations. As mentioned earlier, this guidance is a combined effort of cross-disciplinary scientists. The speakers today are many subject matter experts in stats and quantitative methods and modeling from either Office of Translational Sciences or Office of Generic Drugs. This draft guidance covers recommendations on various topics such as missing data, adaptive design, and specific situations like narrow therapeutic index drugs. It also provides guidance to sponsors and applicants using equivalence criteria in analyzing in vitro or in vivo BE studies. The guidance emphasizes statistical approaches for BE comparisons and explains how to use these approaches in general and specific situations. We have received a total of 101 comments from 12 different organizations, including AAM and stakeholders in industry. We thank all of you for your constructive comments, and we will address each of them with our dedicated effort. Finally, I wanted to state that this draft guidance is also being developed in accordance with the ICH guidelines and other FDA guidances. However, the clear path for some topics, like bioequivalence using multiple references, still under development. The finalized content will replace the 2001 guidance on the same topic. With that, I will pass over the talk to Dr. Stella Grosser to give a further overview on the content of the guidance. Thank you very much.
Hello, everyone, and thank you for the nice introduction. As Liang said, I'm here to give you an overview of our new guidance and describe the contents. The whole need for this guidance is to describe how we can compare test and reference drug products to establish bioequivalence. Bioequivalence being the comparable rate and extent of bioavailability at the site of action. Statistically, these assessments rely on a criterion or possibly criteria, a confidence interval for the criterion, and some predetermined limit for concluding bioequivalence. This is how we have established our decision process. So the guidance, as I said, describes ways to statistically compare test and reference drug products. It also encourages discussion with FDA as methods and technology and the science evolves. There are several pathways such as controlled correspondence with the pre-ANDA, pre-IND, or pre-NDA process pathways that you can take advantage of to talk to the agency early and I hope often. Our guidance includes most topics from the 2001 guidance, plus some additional innovative approaches or innovative technologies that pose new statistical challenges. The table of contents has 4 big sections: the introduction, some general considerations, specific situations, and then part 4, which is listed as part 5, is a set of several technical appendices. Under general considerations, we describe generally study design, data preparation, and statistical models that can be used in the assessment of bioequivalence. First, study design. Well, that constitutes experimental design, and again, these are generally very broadly laid out. There's non-replicated or replicated crossover designs for PK studies. Adaptive design, and this is an innovative topic that we will discuss in greater detail in this webinar. And also problems of sparse sampling designs. There are some indications, some products where multiple samples are not possible to be gathered, to be collected, and that poses its own statistical challenges. And throughout all these designs, the question also arises of sample size determination. As designs get more complicated, it's harder and harder to use canned programs to determine the sample size. And so we encourage sort of scientifically valid and informed ways to determine sample size. Another section in the general considerations part is on data preparation. Here we're talking about log transformations and other data transformations, primarily log transformations. And what's interesting here, and there are these little gems thrown in throughout the guidance, but this is a sort of almost throwaway remark that's important. There are things like, considerations that limited sample size in a typical bioequivalence study precludes reliable determination of distribution. So log transformation or other data transformation should be made based on what is known about the process and the science underlying your product. You need to worry about missing data, intercurrent events, and acknowledge, explain, describe any patterns or reasons for data that are missing, for events that happen after randomization. And there's also the perpetual question of outliers. And that's discussed in this section as well. Another consideration are statistical models. And again, very generally, we are trying to test hypotheses. That you may have heard of the 2 one-sided test, to test the null hypothesis that the products are not equivalent. We implement this approach with confidence intervals, using mixed effects or 2-stage linear models. Again, confidence intervals, there's little bits of important thoughts put in, little bits of wisdom. You need to have equal tail confidence intervals for a valid method, for a valid comparison. The mixed effects models allow for subjects to be correlated so that your reaction to the test is likely correlated with your reaction to the reference drug being considered. The second and largest part of the guidance are on specific situations. There's in vitro situations, pharmacokinetic types of questions, pharmacodynamic, comparative clinical endpoint studies, and related adhesion and irritation studies for transdermal systems. And these will be discussed in detail. Many of these specific situations that call for their own set of approaches will be discussed in today's webinar. But wait, there's more. So stay around and my colleagues will continue to discuss these approaches and this new guidance, new draft guidance. Thank you very much.
Thank you for that introduction. My name is Sung-woo Choi. I'm a mathematical statistician in the Office of Biostatistics and the Center for Drug Evaluation and Research at FDA. In this presentation, I will talk about the statistical test for evaluating population bioequivalence in the updated FDA draft guidance. I will start with brief background information, followed by FDA's population bioequivalence criterion. Then I will discuss the choice of population bioequivalence limit and statistical test for population bioequivalence. As a background, there are two major changes to population bioequivalence in the updated draft guidance. First, we removed individual bioequivalence approach because the individual bioequivalence approach is not currently used in the regulatory setting. The second change is a statistical design. In 2001 guidance, population bioequivalence method was based on replicated cross-off design, but in the updated guidance, we recommend a parallel design is used. Practically, population bioequivalence approach is mainly used for in vitro bioequivalence studies, for example, nasal drug products or oral inhalation drug products. Now we discuss population bioequivalence criterion. Population bioequivalence is assessed using the following hypothesis. Here, theta is defined as population bioequivalence criterion, and theta p is defined as the population bioequivalence limit. Moreover, sigma 0 square is a regulatory constant for variance, and we recommend this regulatory constant is 0.01. We can notice that the population bioequivalence criterion is an aggregated measure, which means including both population means and variances of the test and reference products. Also, we notice that the population bioequivalence criterion is based on a mixed scaled method, which includes reference scaled variance, which is sigma r square, and the constant variance, which is sigma 0 square. Using the sample estimate of reference standard deviation, we can determine which of the two criteria should be used. Now we provide how to recommend population bioequivalence limit, theta p. Population bioequivalence measure can be expressed as the following parts. In the numerator, there are average bioequivalence limit and variance terms. And in the denominator, there is a scaled variance term. An upper B limit of 1.11 is recommended for the average BE limit. Also, FDA recommended allowance for the variance term is 0.01, but this value may be adjusted depending on the average bioequivalence limit for in vitro dataset. Based on these assumptions, FDA recommends population bioequivalence limit, which is delta P, is 2.089. Finally, we provide a statistical test of population bioequivalence in this slide. The population bioequivalence criterion in the null hypothesis can be expressed in terms of the following linear combination of parameters, which is defined as gamma. As I mentioned before, using the sample estimate of reference standard deviation, we can determine which of the two criteria should be used. To provide a statistical test for population bioequivalence, we now need a 95% upper confidence bound for this gamma. Then, the estimated upper confidence bound for that criterion must be less than or equal to 0 in order to conclude population bioequivalence of test and reference products. The formulation of an approximate 95% upper confidence bound can be provided in the updated FDA guidance. This concludes my short presentation. Thank you for your attention. I will be back shortly to answer questions. Thank you.
Hello, my name is Kimberly Rains. I'm a branch chief in the Division of Biopharmaceutics, Office of New Drug Products, Office of Pharmaceutical Quality. Today I will present an overview of IVRT, IVPT, and EMD comparative studies. This presentation reflects the views of the presenter and should not be construed to represent the FDA's views or policies. As a member of the Office of Pharmaceutical Quality, I would like to share a quality product of any kind consistently meets the expectations of the user. Drugs are no different. Patients expect safe and effective medicine with every dose they take. Pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects. It's what gives patients confidence in their next dose of medicine. In the short time I have with you today, I will highlight the content added to the Statistical Approaches to Establishing Bioequivalence guidance, specific situations, in vitro release test, in vitro permeation test, and Earth movers distance probability distributions. This update and revision of the guidance is important to provide clarity and up-to-date recommendations to industry. The presentation can be used for future reference for illustrative examples, literature, and guidance. I will begin by discussing in vitro release test, IVRT. The increasing complexity of drug formulations is driving the application of in vitro bioequivalence testing. The process of using an in vitro technique, such as release testing, to assess a test product's bioavailability and bioequivalence to the reference listed drug product. Here is an illustrated example from literature showing the comparisons of cyclovir products using IVRT. This is not the agency's first introduction to IVRT. IVRT is described in the SUPAC Semi-Solid Dosage Form Guidance, which was published in 1997. It describes the two-stage test for IVRT. In recently published draft guidance, in vitro release test studies for topical drug products submitted in ANDAs, it describes that the assessment of equivalence by IVRT involves a comparison of the median in vitro drug release rates of two formulations using a non-parametric statistical test which is resistant to outliers that are expected to occur under particular testing conditions. I'll now move on to the in vitro permeation test, IVPT. There is a need to develop appropriate methods as alternatives to clinical endpoint studies to determine the bioequivalence of topical products. For semi-solid formulations that differ in excipient composition, in which the partitioning and/or diffusion of an active ingredient into and across the skin may be altered, it is imperative that surrogate in vitro methods be optimized and validated to ensure an effective and reliable determination of bioequivalence is obtained. IVP test is designed to mimic biological conditions. Here's a schematic of the test. Similar to IVRT, IVPT has been described previously by the agency. In the product-specific guidance for acyclovir, the in vitro option describes the statistical conditions needed for a proper IVPT assessment. In the recently published draft guidance, in vitro permeation test studies for topical drug products submitted in ANDAs, it describes that the confidence interval is calculated for each of the endpoints, log transform maximum flux and log transform total amount permeated. The permeation test is performed with excised skin sections for patients undergoing a surgical procedure or from cadaver donors, and the statistical test uses within reference deviation as a threshold that prompts use of either unscaled or scaled confidence interval. Lastly, I'll describe the Earth mover's distance as in the guidance. EMD is a statistical metric that measures the discrepancy between distributions without a prior assumption of distribution. EMD can be used for profile comparison. The EMD is a widely used tool in pattern recognition, machine learning, computer vision, especially for discriminant analysis of histogram-type data. Particle size distribution, PSD, is typical histogram data. The EMD can be used to compare PSD profiles for equivalence tests. When is EMD needed? D50 mainly measures the mean of the profile, and SPAN measures the variability of the profile. However, for complex multimodal PSD profiles, D50 and SPAN may not be appropriate metrics for the profile analysis. EMD comes into play for whole profile analysis. Presented below is a use case in which EMD methodology details are provided for particle size distribution comparison. The agency currently recommends particle size distribution analysis in some of the product-specific guidances. The EMD-based approach has been described in the cyclosporin ophthalmic emulsion product-specific guidance. Thank you for your attention.
Hello, my name is Donald Sherman. I'm an expert mathematical statistician with the Office of Biostatistics in the Center for Drug Evaluation and Research here at FDA. Now, our approach for bioequivalence assessment for both highly variable drugs and for narrow therapeutic index drugs involves the use of reference-scaled average bioequivalence, or RSABE for short. Under this method of assessing equivalence, a test product T and a reference product R are considered bioequivalent if these quantities, in the numerator we have the squared difference between mu T and mu R, which are the population means for the log transformed pharmacokinetic endpoint for the test product and reference product respectively. In the denominator, we have the within subject variance in the population for the log transformed endpoint for the reference product. And we require that this ratio of parameters be less than a constant theta. And we give the theta further structure by specifying these values capital delta and sigma W0. Now, under this criterion, the implied bioequivalence limits on mu T minus mu R, which is to say on the log of the geometric mean ratio, are that difference between means cannot be greater than the log of delta times sigma WR divided by sigma W0. As sigma WR increases, these limits get wider. This differs from regular unscaled average bioequivalence, or ABE, for which the bioequivalence limits are mu T minus mu R has to be less than the log of delta, where typically delta is 1.25, regardless of sigma WR. Now, for highly variable drugs, the center uses capital delta equals 1.25, and sigma W0 equals 0.25. Now, the reference product, having gone through human clinical trials as the basis for its approval, is assumed to be safe and effective. So if the reference product has high within-subject variability, meaning blood levels resulting from it can vary widely from one occasion to another within the same individual, then that product must necessarily have a wide therapeutic window. That argument has been made, for example, by Professor Leslie Bennett in a 2006 presentation before the CEDAR Advisory Committee. So, therefore, there is a scientific justification for defining wider bioequivalence limits for highly variable drugs. Now, in order to estimate sigma WR, the within-subject standard deviation for the reference product, we need a design where subjects receive the reference product more than once. One such design we recommend is what we call the partial replicate design. Subjects are randomly divided amongst 3 treatment administration sequences, and then they each receive the test product once and the reference product twice, in different orders depending on which sequence they're in. Another possible design for highly variable drugs is a 4-period, what we call full replicate design, where every subject receives the test product twice and the reference product twice. Why don't we just recommend the standard 2-period crossover design like we've always used and scale by the residual mean square? Well, 3 factors can make the residual mean square from a 2-period crossover large. If sigma WR is large, yes, but also if sigma WT is large, the within-subject standard deviation for the test product. And it can also be large if there is what we call subject by formulation interaction. Now, of these 3 factors, only sigma WR being large implies a wide therapeutic window, as studied in the original human clinical trials. So we want to scale by sigma WR, and so we must be able to estimate sigma WR. We want to establish that this reference scaled average bioequivalence criterion is less than the constant theta. This implies, with a little algebra, that what is called the linearized criterion is less than 0. So we're going to calculate a 95% upper confidence bound for the linearized criterion and then see whether it's less than 0 in order to establish bioequivalence. The 95% upper confidence bound is calculating using Howe's approximation 1 as described in his 1974 paper. When reference scaled average bioequivalence was being considered for use with highly variable drugs, we realized that while we wanted the implied bioequivalence limits to get wider for high sigma WR, we did not want the limits to get narrower, at least to say narrower than the standard 80 to 125 limits for low sigma WR. For this reason, we formulated a mixed scaling criterion. T and R are considered bioequivalent if the difference between the means is less than log delta, if that's the usual average bioequivalence criterion, if sigma WR is less than or equal to sigma W0, or if the reference scaled average bioequivalence criterion holds if sigma WR is greater than sigma W0. And this is a graph of what the implied limits on mu-T minus mu-R look like under this mixed scaling. We have sigma WR on the horizontal axis and the log of the geometric mean ratio on the vertical axis, and the implied limits remain constant at log 0.8 to log 1.25 until we reach sigma WR equals 0.25. After that, the implied limits begin to get wider. And I would ask you to note that in that graph, there was no discontinuity in those implied bioequivalence limits. So how can we implement the mixed scaling criterion? Sigma WR is unknown, so we don't know whether it's greater than or less than sigma W0. So we have decided to base the choice of statistical analysis on SWR, which is the estimate of sigma WR calculated from the results of the study. If SWR is less than a specified cutoff, we will use regular unscaled average bioequivalence. If SWR is greater than or equal to the cutoff, we will use reference scaled average bioequivalence. And the center has set this cutoff as 0.294, and I note in passing that that cutoff is not equal to sigma W0, which is 0.25. The cutoff of 0.294 for SWR corresponds to 3 decimal places to an estimated within-subject coefficient of variation for the reference product of 30% based on this formula. In addition to the reference scale average bioequivalence or average bioequivalence mixed scaling analysis that we've already described, there is an additional requirement for bioequivalence studies of highly variable drugs, and that is that the point estimate of μt minus μr, calculated from the results of the study, must fall within the usual limits of log 0.8 to log 1.25. Our recommended statistical analysis method for bioequivalence studies of highly variable drugs is described in the draft guidance on progesterone, which was issued in 2012. Now, in the case of narrow therapeutic index drugs, it was noted also by Professor Bennett in that 2006 presentation that narrow therapeutic index drugs tend to have a low within-subject variability. The use of reference-scaled average bioequivalence for narrow therapeutic index drugs was proposed in order to narrow the implied bioequivalence limits for these drugs. The same reference-scale average bioequivalence criterion was adopted for narrow therapeutic index drugs, but with different values for those constants. Sigma W0 is set equal to 0.1, and capital delta is equal to the reciprocal of 0.9, which is approximately 1.11111. We noted that for highly variable drugs, we did not want the bioequivalence limits to get narrower than the standard limits for low sigma WR. In the case of narrow therapeutic index drugs, we do not want the bioequivalence limits to get wider than the standard limits for high sigma WR. For this reason, in addition to the reference scale average bioequivalence analysis, a narrow therapeutic index drug bioequivalence study must also pass standard average bioequivalence with the usual log 0.8 to log 1.25 limits. And we illustrate the implied limits with this criterion in this graph. The implied limits on μt minus μr get wider with increasing sigma WR until it reaches the point where they reach log of 0.8 to log of 1.25, at which point they stop getting wider. In addition to the reference scaled average bioequivalence analysis, the bioequivalence study for a narrow therapeutic index drug must also establish that the ratio sigma Wt over sigma Wr is less than 2.5. Since we must be able to estimate both sigma Wt and sigma Wr in order to test that that criterion is met, we cannot use the partial replicate design for narrow therapeutic index drugs. Our recommended statistical analysis method for bioequivalence studies of narrow therapeutic index drugs is described in the draft guidance on warfarin sodium issued in 2012. And these are the references that I cited, and I thank you for your attention.
Good morning. My name is Feiruz Makhlouf, the Deputy Director of Division of Biometrics 8 in the Office of Biostatistics in Cedar. I will be presenting on the topic of comparative clinical endpoint in bioequivalence studies. In my presentation, I will cover the following topics: why a comparative clinical endpoint study is needed to establish bioequivalence. I will talk about the role of the product-specific guidance in these studies. I will also cover some aspects of the comparative clinical endpoint study, such as study design, endpoints, and analysis populations. I will also talk about the types of hypothesis testing that are used in such studies. There are several ways to establish bioequivalence, such as in vitro testing, in vivo testing in humans, or any other method deemed adequate by FDA. The in vivo testing in humans include bioequivalence studies with pharmacokinetic endpoints, bioequivalence studies with pharmacodynamic endpoints, and bioequivalence studies with clinical endpoints. So when should a clinical endpoint bioequivalence study be conducted? A comparative clinical endpoint bioequivalence study is conducted when it is more informative than the other approaches I mentioned. For example, when the dosage form is intended to deliver the drug locally. Examples of that is topicals such as cream, gel, or ointment, and another example is ophthalmic products. Another reason to conduct a comparative clinical endpoint bioequivalence study is when the substance does not reach the site of action through the systemic circulation. And some examples are the metered-dose inhalers and nasal sprays. FDA usually publish product-specific guidance for generic products to ensure consistency across generic application for the same reference-listed drug. In general, the product-specific guidance outlines the study design, endpoints, study population, and the criteria to establish bioequivalence. The link to all available PSGs are given on the slide. In the next few slides, I will talk about the study design, endpoints, and the analysis population of the comparative clinical endpoint bioequivalence study. First, let's take a look at the study design. The comparative clinical endpoint bioequivalence studies are usually a randomized parallel arm studies with test, reference, and placebo. The test and reference comparison are used to establish bioequivalence. Assay sensitivity, which is the fact that both test and reference products have effects in the study, is determined based on the superiority of both the test and reference products over placebo. The test and reference will be compared separately to placebo. In the comparative clinical endpoint bioequivalence study, we use the modified intent to treat population to assess assay sensitivity. The modified intent to treat population is defined as all randomized subjects who met inclusion and did not meet any exclusion criteria and received at least one dose of products. The per protocol population is used to assess bioequivalence and is defined as all randomized subjects who met inclusion criteria and did not meet any exclusion criteria and received at least one dose of products, and who also met the protocol-specific criteria. The endpoints of the comparative clinical endpoint bioequivalence study could be continuous or binary. Examples of continuous endpoints are mean reduction from baseline in lesion counts and average scale over several assessments. Examples of binary endpoint could be cure or no cure, success, failure. In the comparative clinical endpoint bioequivalence study, we conduct two types of hypothesis testing, equivalence and superiority. To establish bioequivalence, the following compound hypothesis is tested, where the null hypothesis states that the test is either worse than the reference by theta 1 or the test is better than the reference by theta 2. And the alternative is that the test is not worse than the reference by theta 1 and the test is not better than the reference by theta 2. Rejecting the null hypothesis supports the conclusion of equivalence of the two products. This is the two-one-sided test by Sherman. For a continuous endpoint such as the mean of the primary endpoint, the null hypothesis states that the ratio of the means between the test and reference product is less or equal to theta 1, or the ratio of the mean between the test and the reference is greater than or equal to theta 2. The alternative hypothesis states that the ratio of the means between the test and the reference is between theta 1 and theta 2. The null hypothesis is rejected with a type 1 error of 0.05 if the 90% confidence interval for the ratio of the means between test and reference product is contained within the closed interval theta 1, theta 2. Rejection of the null hypothesis supports the conclusion of equivalence of the two products. For a binary endpoint, such as the success rate, the null and the alternative hypotheses are expressed as we see on the slide. The null hypothesis states that the difference of the success rates between test and reference product is less than or equal to delta 1, or the difference of the success rates between test and reference products are greater or equal than delta 2. The alternative hypothesis states that the difference of the success rates between test and reference product is between delta 1 and delta 2. The null hypothesis is rejected with a type 1 error of 0.05 if the 90% confidence interval for the difference of the success rates between test and reference product is contained within the closed interval delta 1, delta 2. Again, rejection of the null hypothesis supports the conclusion of equivalence of the two products. To assess assay sensitivity, the superiority for the test and reference over placebo should be established. As I mentioned before, the test and reference should be compared separately to placebo. So if we let drug A be the test product or the reference product, the null hypothesis states that drug A is not better than placebo, and the alternative states that the drug A is better than placebo. Rejection of the null hypothesis supports the conclusion that drug A, which could be the test or the reference, is superior to placebo. For a continuous endpoint, such as the mean of the primary endpoint, the null hypothesis states that the ratio between the mean of the primary endpoint for drug A and the mean of the primary endpoint of placebo is equal to 1, against the alternative that the ratio of the two mean does not equal to 1. Rejecting the null hypothesis at 5% level of significance supports the superiority of drug A over placebo. For a binary endpoint, such as success rate of the primary endpoint, the null hypothesis states that the difference in success rates of the primary endpoint between drug A and placebo is equal to 0, against the alternative that the difference does not equal to 0. Rejecting the null at 5% level of significance supports the superiority of drug A over placebo. Drug A could be the test or the reference. Finally, this slide summarizes what I covered in my presentation. Thank you for your attention, and I will be happy to take any questions.
Good morning. My name is Wanji Sun. I am from Division of Biometrics 8 of Office of Biostatistics, OTS, CEDAR. I would like to thank the committee for their invitation. The first topic I will discuss today is bioequivalence studies in multiple groups. And this is an overview of what I will cover. First, I will introduce bioequivalence studies with multiple groups. Then, I will discuss bioequivalence assessments for such kind of studies. In particular, I will discuss the treatment by group interaction, as well as what to pay attention in preparing a statistical analysis plan for such kind of studies. Lastly, I will wrap up with a key message. For bioequivalence studies, there can be multiple sources of groups. In new drugs, this is often called subgroups. For example, for certain drugs, due to ethical reasons, patients rather than healthy volunteers are required to establish bioequivalence. For rare diseases or cancers, one clinical center may not be able to enroll enough patients, so multiple clinical centers may be needed in order to recruit a sufficient number of patients to reach the desired power. Patients from different clinical centers may have different severity levels of disease, body weights, or other characteristics. Different clinical centers may have different study conduct. All of this may contribute to different bioequivalence effects across clinics as sometimes observed. This will constitute different groups in a PK bioequivalence study. Especially when there are multiple factors in the study, a combination of different factors may complicate the designation of group. We recommend sponsors to minimize the group effect in PK bioequivalence studies, if possible. For example, all groups should be dosed at the same clinic if feasible. Subjects should be recruited from the same enrollment pool if possible. Subjects should be randomly assigned to different groups and different treatment arms. All groups should follow the same protocol, same criteria, and same study procedures. If feasible, an equal sample size should be assigned to each group. For studies with multiple groups, bioequivalence should be determined based on the overall treatment effect in the whole study population. In general, bioequivalence assessment in the whole study population should be done without including the treatment by group interaction term in the model. However, applicants may use other pre-specified models as appropriate. For complicated scenarios, applicants are encouraged to discuss with the agency via pre-end meetings or controlled correspondence. For studies with multiple groups, the assessment of the treatment by group interaction is very important, especially if any of the first 4 study design criteria previously recommended are not met, and PK bioequivalence data are considered pivotal information for drug approval. If the treatment by group interaction is significant, heterogeneity of treatment effect across groups should be carefully examined and interpreted with care. If the observed treatment effect varies greatly among groups, vigorous attempts should be made to find an explanation for the heterogeneity in terms of other features of trial management or subject characteristics, which may suggest appropriate further analysis and interpretation. It is important to fully pre-specify the statistical methods and models for the primary bioequivalence analysis in the protocol or SAP. If the treatment by group interaction is applicable, detailed statistical criteria and models should be pre-specified. Statistical models should reflect the multi-group nature of the study. For example, period effect nested within group should be used in a crossover bioequivalence study to reflect the fact that the periods for the two groups are different. If there are multiple centers and some have very few subjects, and if the sponsors want to combine centers in analysis, they have to pre-specify the combination rules and the sensitivity analysis is recommended. The take-home message is pre-specify in detail in your statistical analysis plan how to handle multiple groups. The second topic I'll be discussing today is adaptive design for bioequivalence studies. This is new to the 2022 guidance. This is an overview of what I will cover in the next 4 or 5 minutes. First, I will introduce what is an adaptive design. Then, I will discuss the advantages as well as the limitations of an adaptive design. Next, I will discuss how to design and conduct an adaptive study. Lastly, I will give a key message. Now, what is an adaptive design? An adaptive design is a clinical trial design that allows for prospectively planned modifications to one or more aspects of the design based on accumulating data from subjects in the trial. For example, Part 1's Method C is the most popular adaptive design for bioequivalence studies. It is a combination of a group sequential design and an adaptive design with sample size re-estimation. There are also other adaptive designs for bioequivalence, such as Maurer et al.'s standard combination method or maximum combination method. An adaptive design has some advantages over a fixed design. For example, when appropriately implemented, it can provide ethical advantages, increase statistical efficiency or power. It can also reduce resources used and decrease time to study completion. For example, if a study can be stopped early due to establishment of bioequivalence after stage 1, then the sample size will be reduced, the study duration will be shortened, and this will save costs for the applicant. Last but not least, an adaptive design can increase the chance of study success. For example, if a study was underpowered due to underestimation of the variation, an adaptive design can allow reassessment of the sample size after stage 1, and then in stage 2, the sample size will be increased. This will increase the chance of study success greatly. Of course, there is no free lunch. An adaptive design does have limitations. For example, it may need specific statistical methods in order to avoid increasing the chance of erroneous conclusions, such as type 1 error rate, and to avoid introducing bias in estimates of bioequivalence effect. For some complex adaptive designs, such statistical methods may not be readily available. The decision to use or not use an adaptive design is at the applicant's discretion. Now, how to design and conduct an adaptive design? We recommend that the details of the proposed adaptive design should be completely pre-specified and documented in the protocol or SAP. The proposed adaptive design should be scientifically justified, either based on published literature in peer-reviewed journals in which the applicant's proposed approach is validated, or based on extensive simulation results demonstrating desired criteria. For example, the type 1 error rate should be controlled at 0.05 for a bioequivalence test. Applicants should ensure that study integrity will be appropriately maintained. For details, please refer to FDA's Adapt Design for Clinical Trials of Drugs and Biologics Guidance for Industry. For complex adapt designs, we encourage applicants to contact the agency early to discuss their proposed adapt design and statistical methods. The key message is pre-specify your statistical analysis plan and be scientifically justified. With that, thank you for your attention. I'll be back shortly to answer questions.
Good morning. I am Somesh Chakrabarty from the Division of Biometrics 8 in the Office of Biostatistics in CEDAR. In this presentation, I'll talk about the statistical aspects of irritation and adhesion studies, which are a part of generic transdermal product applications. Bioequivalence for generic transdermal products is usually determined using pharmacokinetic studies, and they are analyzed in the same way other pharmacokinetic studies are analyzed. So I am not going to discuss that in this presentation. The studies that are unique for transdermal products are the skin irritation and sensitization studies and the patch adhesion studies. I will first talk about the skin irritation and sensitization studies and then discuss patch adhesion studies. For the skin irritation and sensitization studies, the goal is to demonstrate that the potential for skin irritation or sensitization with the test product is no worse than the reaction with the reference product. So there are two aspects that are studied in such studies: irritation and sensitization. FDA recommends a multi-center, evaluator blinded randomized study in healthy subjects. A skin irritation and sensitization study has two main phases: a 21-day induction phase where skin irritation is evaluated and a challenge phase where sensitization is evaluated. There is a 14 to 17-day rest period between the induction phase and the challenge phase. A re-challenge phase similar to the challenge phase is conducted 4 to 8 weeks after the challenge phase if potential sensitization reactions are observed in the challenge phase. In the induction phase, both test and reference patches are applied simultaneously, and the patches are replaced by new patches of the same type at the same locations for repeated applications. For both induction and challenge phases, the test and reference patches are applied on two sides of the body. For example, if the test patch is applied on the left arm, the reference patch is applied on the right arm. The sides are randomized for each subject. The wear duration is determined based on the reference product's label. The skin location is assessed for irritation after removal of each patch. In the challenge phase, a single test and a single reference patch are applied simultaneously at 9 sites that were not used in the induction phase and kept for 48 hours. Irritation assessments are based on an 8-point dermal response scale and a 4-point other effects scores. For each of these, a higher score reflects a higher level of irritation. Irritation score is the sum of the dermal response and the other effects scores. In the induction phase, each patch application site is assessed for irritation after removal of each patch. In the challenge phase, 4 assessments for skin reaction are made after a patch is removed. They are at 30 minutes, 24 hours, 48 hours, and 72 hours after the patch removal. The primary endpoint for irritation analysis is the mean irritation score. A non-inferiority analysis is conducted using a non-inferiority margin delta of 0.2. The null hypothesis is that the mean irritation score for the test product minus the mean irritation score for the reference product, that is mu t minus mu r, is greater than or equal to the non-inferiority margin delta. and the alternative hypothesis is that mu t minus mu r is less than delta. The test is performed at one-sided 5% level of significance. Non-inferiority of the test product to the reference product in terms of irritation is established if the upper limit of the 90% confidence interval for mu t minus mu r is less than 0.2. A mixed effects model is used for the irritation analysis, that is to construct the confidence interval for mu t minus mu r. The study should have a sufficient number of subjects for the power to be 80% or more. The per protocol population is used as the primary analysis population. The per protocol population for irritation and the per protocol population for sensitization are defined per patch type per subject. Details of these populations can be found in FDA's draft guidance on irritation assessment. There are other secondary endpoints that you can find in the draft guidance as well. The sensitization analysis is descriptive. For the adhesion studies, the goal is to show non-inferiority of adhesion characteristics of the test product with those of the reference product. For an adhesion study, FDA recommends a single-dose randomized 2-treatment, 2-period crossover design. Each subject is randomly given one of the test and reference patches in period 1 and the other patch in period 2. Alternatively, a single period 2-treatment per subject design may be used with a proper justification. In that design, both test and reference patches are applied simultaneously in a single period, and the treatments are randomized between the application sites for each subject. Often, pharmacokinetic and adhesion evaluations are done in the same study. The second design cannot be used in those studies. Generally, the study should be conducted in healthy subjects. Multiple adhesion assessments are made throughout the duration of wear. Adhesion assessments are based on a 5-point adhesion scale, 0 to 4, which is given in this table, where 0 indicates at least 90% adhered and 4 indicates complete detachment. Note that the percent adhered is not equally spaced between the boundaries of the numeric scores. The primary endpoint in the adhesion analysis is the mean adhesion score, which is defined for each patch as the mean of all adhesion scores post-baseline. If adhesion assessments are not equally spaced in time, a weighted mean should be used, where the weight for an adhesion score is the time between that assessment and the preceding assessment divided by the total planned duration of wear. At any time point, the highest adhesion score up to and including that time point is used instead of the adhesion score at that time point in the calculation of the mean adhesion score. This is because any decrease in adhesion score over time is due to some external influence such as external pressure on the patch and cannot be attributed to the adhesion properties of the patch. The null and alternative hypothesis and the primary analysis of adhesion are very similar to those that you have seen for the irritation analysis. However, the non-inferiority margin for the primary adhesion analysis is 0.15. Non-inferiority of the test product to the reference product in terms of adhesion is established if the upper limit of the 90% confidence interval for the mean adhesion score for test minus the mean adhesion score for reference is less than 0.15. Here are the two references. FDA's draft guidance on assessing the irritation and sensitization potential of transdermal and topical delivery systems for ANDAs, and FDA's draft guidance on assessing adhesion with transdermal and topical delivery systems for ANDAs. Thank you for your attention.
Hello everyone. This presentation will focus on dose scale analysis and how it can be used to support bioequivalence assessment. This presentation reflects the views of the speaker should not be considered to represent the advice or guidance on behalf of the US FDA. This is the outline of this presentation. First, I will give a brief introduction what pharmacodynamic PD equivalent studies, and then I will introduce dose scale analysis for PD studies, including what it is and when to use it, as well as the recommendation to use it. Lastly, the considerations for model fitting methods and the bootstrap implementation will be also discussed. To establish therapeutic equivalence of generic drugs to its reference product, generic drugs need to demonstrate pharmaceutical equivalence and bioequivalence, BE. BE is defined as no significant difference in the rate and extent of absorption between a test product and the reference product. To establish BE, usually 4 types of studies can be used, including comparative pharmacokinetic BE studies, comparative PD BE studies, comparative clinical endpoint studies, and in vitro BE studies. Today, we will talk about comparative PD BE study. For some drug products, especially locally acting drug products, PK studies may not be sufficient, so PD studies are recommended to support BE establishing. In current product-specific guidance, PSG, PD studies have been recommended for oral inhalation drug products, locally acting GI products, and topical corticosteroid. This slide highlights the difference between BE based on the PK and the BE based on the PD endpoints. As shown in the left panel, for BE based on the PK endpoints, the exposure is proportional to the dose. There is no exposure for placebo, and the 90% confidence interval around exposure ratio can be used for BE. For BE based on PD endpoint, as shown in the right panel, it shows nonlinear dose response. That is, response does not increase proportionally with the dose. In addition, placebo effects can be large, and the 90% confidence interval around the PD response ratio often should not be used for BE. Given the nonlinear dose-response relationship, the difference in PD response may not directly reflect difference in the relative bioavailability of the test and the reference product. To address this issue, dose scale analysis was introduced to allow the assessment of relative bioavailability on dose scale, not original scale of PD response. That is, transforming nonlinear PD scale to linear dose measurement scale, so the results can be used to support equivalence of the amount of drug reaching the site of action. As shown in the left side panel, there are several components for dose scale analysis. First, we will have a response from the placebo as a baseline, and then PD response from the reference product at the lower dose and higher dose, respectively, shown as 3 small gray squares in the figure. From this, we can fit a dose response curve for a reference product. Then we have a test PD response at a lower dose. In this example, it's about 90 microgram. This test PD response is shown as a small blue square in the figure. From the dose response curve for reference product, we can find the estimated dose for reference product that would result in the PD response equal to that from the test product. In this case, it's around 100 microgram, shown as a small red dot in the figure. Then, the relative bioavailability F can be calculated from the estimated dose, which is 100 microgram, divided by the actual dose, which is 90 microgram. So in this case, the relative bioavailability F is 1.11. The essence of dose scale analysis is the Emax model fitting. Due to the time limit, I will not go through all the details about this equation. The detailed information can be found in the link below. Here, I just want to emphasize a few points for this analysis. This model is based on assumption that both E0 and Emax are the same for the test and reference products. Second, when applying the nonlinear mixed effect model, the fixed effects are E0, Emax, ED50, and F, and the between subject random effect should be specified for the parameters such as E0 and Emax, and the residual error random effect should be included as well. For Emax model fitting, there are several methods available, including naive average data, naive pooled data, and nonlinear mixed effect modeling. For naive average data, data from individuals are averaged, so there are only one data point for each dose for each formulation, as shown in the left figure. For naive pooled data, data from all individuals will be pooled as if they're coming from one single individual, as shown in the middle figure. For nonlinear mixed effect modeling, all individual data will be used for modeling while incorporating the between subject variability. As a quick summary, naive average method actively reduces available observation. There is no direct estimate of variability. This method is biased if between subject variability is large. It's also potential biased if individuals have a different amount of data or aberrant observation. The naive pooled method is preferable to naive average method. However, it's also biased if between subject variability is large. In addition, it introduces potential bias as data coming from non-standard designs can be put together. Compared to above two methods, nonlinear mixed effect model is recommended because the model is capable of characterizing between subject variability and the residual unexplained variability, and also less sensitive to aberrant observation and missing values. To establish BE, 90% confidence interval for F should be calculated. Two methods are recommended. The 90% confidence interval can be directly obtained from the nonlinear mixed effect model, which is directly from the point estimate of log F and its standard error calculated using the model. Alternatively, the 90% confidence interval can be obtained from the bootstrap procedure, which includes three steps. First, generating sample dose response datasets using bootstrap sampling with replacement. Second, estimating F by feeding the Emax model to each sample dose response dataset. 3. Computing 90% confidence interval for F using Efron bias corrected and accelerated method. For bootstrap sample, we have seen various ways to generate sample dose response dataset. Here, I'm showing an example of different ways to conduct bootstrap sampling. From left to right, the first table is the original data with 3 subjects in a crossover study. The second table shows bootstrap sample by resampling observation without reserving subject information. The third table shows the bootstrap sample by resampling subjects but only for reference data. The fourth table shows bootstrap sample by resampling subjects. Among 3 resampling methods, we recommend the last one, that is, bootstrap sampling unit should be the subject, and remaining all the data from test and reference in order to maintain the correlation of the observations within subject. Here are some practical considerations. For fitting Emax model, nonlinear mixed effect model is preferred because it incorporates between subject variability and less sensitive to aberrant observation. For modeling software, non-man, SAS, R, and etc., results are generally consistent with the same model structure and parameter setting. For computing 90% confidence interval of F using bootstrap, resampling original dose response observation at the subject level should be used. Minimum of 1,000 bootstrap are typically needed. We recommend following the bootstrap procedure in the PSG. and the applicant should pre-specify modeling software and the computation method for 90% confidence interval. As a summary, the dose scale analysis has been used to demonstrate PD equivalence for locally acting drug products with a nonlinear dose-response relationship. And FDA's guidance reflects agency's current thinking and recommendation. Towards reliable dose-scale analysis, applicants should have appropriate planning, including pilot study for your study, and state how missing data will be handled in the protocol, and also provide sufficient justification for alternative approaches that are not in the guidance. Applicants are encouraged to discuss significant difference or alternative approach with OGD. That concludes my presentation. Thank you.
Good morning. You have already heard about excellent presentations on the 2022 revised BE statistical guidance. In the next 10 minutes, I will be sharing with you some additional key recommendation updates from the revised guidance and how they are connected to our BE assessment. First, I will give a brief introduction of BE studies assessed by the Office of Bioequivalence. Next, I will discuss some recommendations in the revised guidance, including model options, group analysis, outliers, and modeling and simulation-based approach. By the end of the presentation, I hope that you will have a better understanding of these important updates and how they can be applied to your work. The Office of Bioequivalence reviews most BE studies, including pharmacokinetic BE studies, pharmacodynamic BE studies, and in vitro BE studies. PKBE studies include two-way crossover study, partially replicated study, fully replicated study, narrow therapeutic index drug study, parallel study, sparse sampling study, abuse deterrence study, etc. PDBE studies include vasoconstrictor study. A revised guidance was published in 2022 on this topic. PDBE studies also include bronchoprovocation study and fecal fat excretion study, both of which are analyzed using dose-scaled analysis. Mon just covered this topic. In vitro BE studies include in vitro binding study, in vitro release test, in vitro permeation study, particle size distribution, recovery, spring pattern, plume geometry, comparative dissolution. Some of these topics were already covered by my colleagues today. The Office of Bioequivalence does not completely rely on applicants' statistical results to make a BE determination. Instead, we conduct independent statistical analysis for all BE studies mentioned here. The revised BE statistical guidance includes model options for applicants to choose. I list some examples here. In a two-way crossover study, applicants can use a general linear model, GLM, or mixed model to conduct statistical analysis. The GLM model removes all subjects with any missing observations for any variables included in the GLM model. But the mixed model uses all observed data. This difference could lead to different BE conclusions, so it is important for applicants to pre-specify their statistical model. Similarly, for sparse sampling, applicants can choose bootstrap or parametric methods for estimation of standard deviation and the confidence interval for the ratio of AUCT. For dose-scaled analysis, applicants can choose resampling or nonlinear mixed effect model without resampling. In the PROC mixed procedure, applicants can choose to use either FA02, CSH, or UNR in random statement, and choose to use centers or KR2 as denominator degree of freedom in the proc mixed procedure. Again, the most important message is that the selection of option could lead to different BE conclusions, so it is important to pre-specify your statistical model. Data-driven post-hoc selection of the statistical model is not allowed. Wanjie has given a comprehensive presentation on group analysis. Since group analysis is a major update in this revised guidance, I will emphasize it again. Statistical methods and models should be pre-specified in details in the protocol or study analysis plan. BE should be determined based on the overall treatment effect in the whole study population. Appropriate group terms should be included in the statistical model. Subgroup analysis is only used as sensitivity analysis if there's a significant group effect. The significance of group effect is determined by the p-value for the treatment by group interaction term in the average BE analysis, or the p-value for the group term in the reference scaled ABE analysis. The reason for large heterogeneity among groups should be investigated. Pulling smaller groups into a larger group may be acceptable if justified and pre-specified. Sensitivity analysis on different pooling methods is recommended. Another update in the revised BE statistical guidance is about outliers. In general, outlier data may only be removed from the BE statistical analysis if there is real-time documentation demonstrating a protocol violation. The existence of a subject outlier with no protocol violations and for which there are no bioanalytical errors could indicate product failure or subject by formulation interaction. All subject data should be submitted with potential outliers flagged with appropriate documentation as part of the submission. In general, data with outliers that have no protocol deviation or bioanalytical errors are used for the BE determination. A sensitivity analysis is also useful to evaluate the impact of outliers on the BE conclusion. For example, for a replicated PKBE study, if reference scaled average BE is used, the applicant should ensure that the calculated intra-subject variability is not inflated due to extreme values or situations. The revised BE statistical guidance includes the recommendation of modeling and simulation-based approach. It states that modeling and simulation-based approaches may be utilized in some scenarios, such as sparse sampling studies or missing samples. A modeling and simulation-based approach should be pre-specified. A post-hoc modeling and simulation-based approach cannot be used to salvage a failed BE study. That is, it is not acceptable that you conduct a regular statistical analysis and find the study fails, and then you use a modeling and simulation approach to show that the study could pass. Overall, the key message to you is that please pre-specify your statistical analysis plan in details. If you have any questions about your statistical plan, please contact the agency before you start your study. That concludes my presentation. I will be back shortly to answer questions in the Q&A session. Thank you.
Thank you to all of our speakers for the great presentations. Moving into our Q&A session, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. It's our pleasure to welcome to the Q&A panel Dr. Lucy Fang, Deputy Director, Division of Quantitative Methods and Modeling, Office of Research and Standards, OGD, Dr. Ying Fan, Lead Pharmacologist, Division of Clinical Review, Office of Safety and Clinical Evaluation, OGD, and David Coppersmith, Regulatory Counsel, Office of Generic Drug Policy and OGD. Looks like we do have some questions already coming in right now. And the first question is directed to Dr. Lucy Fang. And here is the question. For BE studies with 2 reference treatment arms, can we only submit BE data on the test product and the U.S. reference product?
Thank you for the question. The B studies, the B analysis should be conducted comparing the test product and the U.S. reference product. However, the complete data, including the test product, which is the subject of the application, as well as the non-U.S. reference product, should be submitted for the completeness. Thank you for the question.
Thank you for responding to that question. Moving on to our next panelist, we do have a couple questions that came in for Dr. Ying Fan, and here is the first question. Can you provide some explanation from a clinical perspective how you determine the analysis populations, in particular the per protocol populations for the comparative clinical endpoint bioequivalence studies in ANDAs?
Thank you for the question. From a clinical perspective, we will review multiple places in the submission, such as subjects' medical history, concomitant medications, subjects' inclusion-exclusion criteria, and also protocol deviations, variations, adverse events, and also like reasons for per protocol population exclusion, and also reasons for subjects discontinuations, and to see if the subjects are appropriately included or excluded in the per protocol population for the bioequivalence analysis. And if there is any subject that should not be included or excluded, due to medical history, inappropriate concomitant medications, or the reasons for exclusion is not appropriately, then we will make adjustments to the per protocol population accordingly. So we do recommend that you record the medical history, concomitant medications, and the subject discontinuation reasons, and also like population exclusion reasons carefully, and also in detail when you conduct the clinical endpoint bioequivalence study, and also submit all the information that you have in original ANDA submission. So to avoid, receive the information request in the review cycle. Thank you.
Thank you for responding to that question. We have another question that just came in for Dr. Fan, and here is the question. In the current statistical bioequivalence guidance, it is stated that for the statistical analysis approach for irritation, sensitization, and or adhesion studies for transdermal and topical delivery systems, refer to the guidance for general irritation, sensitization guidance, and general adhesion guidance published in 2018. Can you let us know if there is going to be a revision for these 2 general guidances?
Thank you for the question. Currently, the general irritation and sensitization study guidance and adhesion guidance are under the revision process. So we are carefully visiting all the comments that we received through the public docket since these two guidance were published in 2018. So we recommend that you pay attention to the FDA website for the most updated information regarding these two guidances revisions. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we do have a question that just came in for David Coppersmith. And here is the question. The most recent version of the guidance was issued as a draft guidance. When will FDA issue a final guidance?
Thank you for the question. The draft guidance was issued for comment, and as Dr. Zhao mentioned during his introduction, FDA received several comments during the comment period. FDA is reviewing the comments received through the public docket and will prepare either a final version or a revised draft version of the guidance that incorporates changes when appropriate. When finalized, this guidance will replace the February 2001 guidance of the same name and will represent FDA's current thinking on this topic. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we have a few questions that came in for Dr. Sungwoo Choi, and here is the first question. Could you please elaborate why the study design recommendation has changed from crossover to parallel? Dr. Choi, we are not hearing any audio from you right now. What we're going to do is go on to our next panelist, let our tech team reach out to you to adjust your volume, and then we'll check back in with you later on. Moving on to our next panelist, got a few questions that came in for Dr. Kimberly Rains, and here is the first question. Is the IVPT BE stats guidance applicable to the TDDS patches, which are intended for systemic use in the same manner as a semi-solid topical preparation? Sorry, Dr. Rains, looks like we're having a little bit of audio problem right now. We can't hear you, so we're going to go on to move back up to Dr. Choi to see if they're able to adjust the volume and the audio connectivity. And so we're going to move back to Dr. Choi. And here is the repeat question for Dr. Choi. Could you please elaborate why the study design recommendation has changed from crossover to parallel?
Hi, thank you for the question. The population bioequivalence method using crossover design is not statistically valid because this method does not meet one of the main statistical assumptions, which is independence assumption in calculating the upper confidence bound. And another reason is that the population bioequivalence is currently recommended to use for many in vitro bioequivalence, and in vitro bioequivalence method is based on a parallel design. This is why the study design recommendation has changed from crossover to parallel design. Thank you.
Thank you for responding to that question. We do have another question for Dr. Choi, and here is the question. What is the difference between population BE and a normal BE study, and in which scenario is a population BE required?
Thank you for the question. Population bioequivalence requires that both the means and variances between the generic and reference products to be sufficiently close, but average bioequivalence, he mentioned the normal bioequivalence, but I assume this is average bioequivalence, only requires the means between reference and genetic products to be sufficiently close. And as I mentioned before, population bioequivalence is recommended for in vitro bioequivalence studies in case of very less variance. In other words, the variance is very small, and in case of being more sensitive indicators of drug delivery than the in vivo bioequivalence studies. So currently, most oral inhalation drug products and the nasal drug products recommended using population bioequivalence for in vitro bioequivalence method. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions that came in for Dr. Donald Sherman, and here is the first question. How is a highly variable drug defined, and is this information available on and within the RLD labeling?
This is Donald Sherman. I want to preface my response by saying that anything I say, not just in response to this question, but any question I address in this question and answer session, anything I say represents my personal opinion and does not necessarily reflect the views of the Food and Drug Administration. A fairly well-known and well-established criterion for what constitutes a highly variable drug product is a within-subject coefficient of variation of 30% or more. I'm not the one to address whether that would be reflected in the labeling, but it would require that crossover studies be done on the original reference listed drug, and I would doubt that usually would be the case.
Thank you for responding to that question. We do have another question for Dr. Sherman, and here is the question. If the full replicate study of some subjects are completed, the partial replicate arm like RTR or TRT, is it feasible to include these subjects in the final analysis for calculating the 95% upper bound?
The short answer is no. The methods that we have established for doing the analysis with a fully replicated design would require all of the subjects to be treated the same. And while how to do a highly variable drug reference scaled analysis in the presence of missing values is a current topic of research and is very much of interest, I am not aware of any validated methods to include those subjects.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions that came in for Dr. Feroz Makhlouf. And here is the first question for Dr. Makhlouf. Observe that there is a different PSG published on the US FDA and the EMA. Please clarify the difference between the two, and in what situation or scenario should one regulatory agency be followed versus the other?
Hello, thank you for your question. So if the intent is to submit to FDA, it is advised that the sponsor should contact the agency directly with questions regarding the study design. I cannot comment on, EMA versus FDA in the product specific guidance. Thank you.
Thank you for responding to that question. We do have another question for Dr. McLuth. And here's the question, and it's regarding on comparative clinical endpoint bioequivalence studies. What happens if equivalence to the reference could be demonstrated and one runs in the borderline scenario that test is superior over placebo and reference is not superior over placebo?
Thank you for this question. Also, as I mentioned in my slide, that to assess the assay sensitivity, both the test and reference products should be superior to placebo. So in this case, if the reference fails to show the superiority, then the assay sensitivity is not established. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a couple questions that came in for Dr. Wanji Sun. And here is the first question for Dr. Sun. And it involves whether it is required to evaluate separate ANOVAs for each group to evaluate treatment heterogeneity.
Oh, hi. Thank you for the question. So as we said, the bioequivalence should be based on the overall bioequivalence test among the whole study population. However, there's assumption behind this. We're assuming that there is homogeneous treatment effect across different groups when there are multiple groups in the PK bioequivalence study. So if this is not true, then we cannot really estimate an overall treatment effect. So if the interaction term between study and the treatment is significant, which means that the treatment effect is not the same across different groups, then that's a flag that we have to pay attention. So in that case, we need to look at the treatment effect in each group and then see whether the interaction term is what we call a qualitative interaction term or a quantitative interaction term. So we recently published a paper in Statistics in Biopharmaceutical Research. So qualitative versus quantitative treatment by subgroup interaction in equivalent studies with multiple subgroups. So you can refer to that paper for the definition and also the cutoff to detecting different interaction terms. So short answer is yes, you need to look at each group and to look at whether the difference is large enough that we cannot actually combine the two groups into to evaluate an overall treatment effect. Thank you.
Thank you, Dr. Sun, for responding to that question. We've got a couple more questions that came in for you, and here's the next question. If the study is planned without adaptive design, but due to low subject availability, the study was concluded with available study data, and the study met the BE criteria comfortably, does the applicant still need to check for type 1 error?
So that's a very good question. So for adaptive design, we have to say that the sponsors need to pre-specify a adaptive design if they plan to use one in the protocol or SAP prior to the conduct of the study. So with that said, the sponsors are not supposed to do a interim analysis after collecting just the partial data and evaluate bioequivalence. So this is not allowed. So for an adaptive design, if you plan to do an interim analysis, as we said, you need to pre-specify that. So I hope that addressed that question. Thank you.
Thank you for responding to that question. We have one more question for Dr. Sun, and here is the question. Are Potvin's methods appropriate for BE studies in the regulatory context? It has been mentioned in some literature that Potvin's methods may lead to type 1 error inflation by simply combining individual data from two stages.
Yeah, that is a very good question. So Potvin's methods currently are the most popular adaptive design methods for bioequivalence studies. So in Potvin's paper, 2008 paper, the authors did extensive simulation to evaluate the type 1 error rate inflation under different scenarios. However, because Potvin's methods are based on simulation, which means that it is impossible to exhaust all the possible scenarios, what we call a sample space in statistics. So there are scenarios that are not covered in the paper and are not covered by the author's simulation that may, there is a slight type 1 error rate inflation. So that's why there are later papers that use a more stringent, analytical method to guarantee that type 1 error rate is covered under 0.05, so-called the p-value combination method, so which has been used very often in new drug. So with that said, Potvin's methods is appropriate for bioequivalence studies, and we did do extensive simulations later on. There was a poster that 2 years ago at APS. We have a visual tool that shows vividly in which, most of the scenarios, most of the regions are safe. Majority of the regions are safe, but there are, small regions where type 1 error rate could be inflated over 0.052. So you can refer to that poster for that. But overall, Potvin's method is appropriate for bioequivalence studies, but just pay attention to those pitfalls. Thank you.
Thank you for responding to that question. Moving on to our next panelist. We've got a question that came in for Dr. Sumesh Chattopadhyay. And here is the question for Dr. Chattopadhyay. If the reference listed drug is highly adhesive, for example, an average adhesive score of 0, will it not be more difficult to establish non-inferiority of the test product to the RLD?
Hello, this is Sumesh Chattopadhyay. If it is actually in this scale, the difference that we are looking at, basically the mean adhesion score, the difference between the mean adhesion score in the test arm test and the reference product, it is not going to affect the how adhesive the reference product is. If the if the evaluation was based on the ratio, then it would be, but in this case it is just the difference, so it is not going to be a problem. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a question that came in for Dr. Ming Hu, and here is the question for Dr. Hu. Orlistat is a fecal fat excretion. The current FDA guidance BE limit is 80 to 125%. The fecal fat excretion is variable. Is it feasible to widen this acceptance limit?
Thank you for the question. This question is very interesting, but also complicated to answer right now. We may not touch upon all the details for this question, especially the modeling and the simulation are involved. So I will suggest you kind of communicate with us via control correspondence or pre-ANDA meeting. Then we can get started from there. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a question just came in for Dr. Zhang, and here is the question for Dr. Zhang. Can we use R-based code for B analysis as a replacement to SAS?
Yeah, thanks for your question. Yes, you can use R-based code or any other software for BE analysis as long as your code is validated. Thank you.
Thank you for responding to that question. We've got a few more questions coming in, and we're going to try to see if Dr. Rain's audio is working okay. And here's the question for Dr. Rains. Although not mentioned in today's talk, what are the bioequivalence statistical considerations for in vitro abuse deterrent formulation comparative studies?
Thank you for the question. The general BE statistical considerations for abuse deterrent formulations can be seen at the abuse deterrent for opioids and evaluation of labeling guidance that the agency has also produced. Things that are examined are the potential routes of abuse, injection, or if it's inhaled. And
Dr. Rain, sorry we're not able to hear you at this time. This is the moderator coming back on. We're going to move on to one of our next panelists and then let our audio experts reach out to you to see if they can adjust your volume so we can get another question into you. And just as more questions are coming in, remember if you haven't, as we just got a few minutes left, if you have an urgent question, please submit that question now. Looks like we have another question here for Dr. Zhang. And here is the question. What happens if review teams obtain different statistical results from applicants when conducting their own statistical analysis?
Thanks for the question. The review team's action depends on different scenarios. For example, if the review team can figure out what causes this discrepancy in statistical results, and if this discrepancy does not lead to opposite bioequivalence conclusion, then the review team will not issue a deficiency for this discrepancy. Otherwise, usually the review team will ask applicants to provide more information. Thank you.
Thank you for responding to that question. We just have just a little bit over a minute left in our remaining time. We're going to try to get one more question in for Dr. Sungwoo Choi. And here is the question. Why is population bioequivalence method recommended to use for many in vitro BE evaluations of orally inhaled and nasal drug products?
Thank you for the question. As I mentioned before, population bioequivalence recommended for the many in vitro bioequivalence studies because in many in vitro bioequivalence studies, for example, nasal drug products or oral inhalation drug products, is a very less variance. In other words, the variance is sometimes really very small, and also this product is more sensitive indicators of drug delivery than comparing with the in vivo bioequivalence studies. So that's why we currently population bioequivalence is recommended for in vitro bioequivalence studies, such as the oral inhalation drug products or the nasal drug product. Thank you.
Thank you for responding to that question, and then we want to thank everyone for their very informative and timely talk, and also for responding to numerous questions that came in because we're just out of time. We do have a few closing reminders. This activity is eligible for an attendance certificate which supports continuing education by SOCA RAPS SQA and ACRP. Please be sure to refer to our website at fda.gov/CEDAR-SBIA for more details. You'll only be able to obtain the attendance certificate upon completion of the survey, which remain open for 2 weeks. And remember, that's March the 28th. Please remember to download the certificate immediately upon completing the survey. It cannot be retrieved after the fact. Let us know your comments and feedback via the survey. This will help guide the design of our future events. Additional tutorials are available through fda.gov/CEDAR-Learn. On behalf of CEDAR Small Business and Industry Assistance, we hope that you enjoyed today's presentation and found it helpful. We look forward to your presence at future webinars, which will be advertised on our website at fda.gov/CEDAR-SBIA. Thank you for attending, and this concludes today's webinar.